So now we will be discussing about cell culture. So cell culture actually refers to the removal of the cells from an animal or plant and then subsequently growth in some artificial environment. Definitely the environments need to be suitable for their growth. So the cells can be removed from the tissue, directly from the tissues, which is called the primary cell line. And then they are actually disaggregated. That means normally they just clump together. So we need to dissociate the cells by mostly the enzymatic treatment, and some mechanical means can also be used before the cultivation. They can be derived from a cell line or a cell strain that is already been established, which is also called the secondary cell line. So the culture conditions vary widely for each cell type, but the artificial environment where the cells are actually cultivating is more or less similar and consists a suitable vessel containing a substrate or the medium that actually supplies the essential nutrients for the cell, such as amino acids, carbohydrates, vitamins, and minerals. Also, the growth factors and hormones, and all the cells also need the gases, oxygens, and carbon dioxide, as well as some physiochemical environment like the pH or the osmotic pressure and temperature. Most of the cells are really anchorage dependent. That means they require a vessel and the substrate where they actually attach to them, and they normally form a monolayer of a culture. However, there is other cells type is also there, which can be growing while they are floating in the culture medium. So that is like a suspension of culture. So the primary culture is actually referred to the stage where the culture of the cells are isolated from the tissues and then proliferated under some appropriate conditions or a suitable environment until they kind of grow all of the available substrate that is, and this growth of the cell is called a confluence. So at this stage, that means when the cell growth reach the confluency, when they sort of utilize all of the substrate, we need to subculture the cell. That means we take some of the cells from that first culture and transfer them into a new vessel with a fresh growth medium, including the nutrients to provide a more room for their growth. And this subculturing is actually called passage. So after the first subculture, the primary cell culture then become known as a cell line or subclone. And the cell lines derived from the primary culture have a limited time lifespan. That is, they are finite. And as they are passaged, cells with the highest growth capacity, they actually predominate into the vessel. Normal cells usually divide only a limited number of times before losing their ability to proliferate, and which is genetically determined event known as senescence. These kind of cell lines are known as finite, just described. However, some cell lines, they can be immortal by a process called transformation, which can be occur by spontaneously or chemically or induced by virally. And when these finite cells are acquiring the ability to divide indefinitely, it can then cause a continuous cell line. So according to the morphology, all of the cells actually divide into three basic types. That's fibroblastics, epithelial-like, and lymphoblastics. So the fibroblastic cells, or the fibroblast-like cells, are bipolar or multipolar, and they have a elongated shapes and grow on attached to a substrate. That is the anchorage, which we described in our last slide. On the other hand, the epithelial-like cells are a polygonal shaped, but their dimensions are more regular or distinct, and they also grow in a substrate, attached to a substrate with a discrete kind of patches. While the lymphoblast-like cells, they are more of a spherical shape, and they usually grow in suspension, like without attaching to a surface. So these are the three basic morphology of a cell culture. So what are the applications of cell culture? Cell cultures is extensively used to understand or decipher the cellular and molecular network of any genes. And it provides an excellent model system for studying the normal physiology or biochemistry of the cells, like the metabolic studies, aging, and it can actually use specific cell to check or investigate specific molecular event, such as you can isolate the cardiac cells from the cardiac tissue of a mice and grow them as a primary cell culture, and that cell can be used to get the signaling or the molecular function of any cardiac genes or any genes that have an effect on cardiac activity. So this cell culture also can be used to understand the drugs, the effects of drugs and the toxic compounds, mutagenesis. You can do mutation to the gene of a specific gene, like you can mutate a neurological gene and use that gene into the neuronal cell culture to understand the effect of that gene in neurons. Carcinogenesis, cancer cells, there are hundreds of cancer cells, has been used all over the world for research to understand the specific cancer type. So you can actually use a lung cancer cell line to understand what is the signaling mechanism in that lung cancer cell. You can use a prostate cancer cell line to understand what is the signaling or the molecular function there. Or you can select a specific gene for that prostate cancer and then mutate the gene and use a prostate cancer cell line to understand the effect of that genes into the molecular network. So you can actually do a lot of research by doing specific cell culture and a specific gene involved in that cell regulation. Drug screening, again, that any drug which can be used for the cancer cell line, which can be used for a cancer like prostate cancer or lung cancer or brain cancer or any other types of cancer, you can specifically use that cell line to investigate that what is the effect of that drug into that cancer cell line. And definitely large-scale manufacturing of biological compounds, such as vaccines and therapeutic proteins. So the major advantage of using the cell culture is that the consistency and reproducibility of the result, or by using the cell culture batch after batch, you can actually get a consistent and reproducible result, and which is very important in any kind of research, including working into the biotech companies. So these are the brief overview of a cell culture and the applications. I have included some of the videos of the cell culture, how to use the cell culture hoop, what are the specific things we need to take care of while we are doing any cell culture experiments. And you can also get, I think, other videos like how to preserve the cell and this kind of thing. What are the lab specific requirements of doing a cell culture experiments? So I hope we will definitely discuss all of this in our discussion class if you have any questions. So thank you.